Okay, the last thing that we're going to learn about on this topic of antigens, antibodies, is antigenic drift. And it's really important to know that this is only going to apply for the flu virus. So we're not talking about antigenic drift when we talk about measles or HIV. It's very specific to flu virus. And it's really important to understand because you'll very logically see why it's important to get your flu shot every single year is because of antigenic drift. Okay, so a little bit of background. It's really important to note that influenza virus, the flu virus, is an RNA virus. So that means that the piece of genetic material that's in the middle is an RNA. And so remember that viruses need the host machinery in order to make more of itself. It can't do that by itself. So the flu virus attaches to a host cell receptor. It enters into the cell, and it needs that receptor to do so. When it gets into the cytoplasm, the envelope and capsid open up and release the piece of RNA genetic material into the host cell. And that RNA, think about how we make more RNA. We have to do that in the nucleus with an RNA polymerase. So what happens is that flu RNA, or viral RNA, has to get into the nucleus and use our machinery to get more of itself. And you may not remember this from your biology class or if you've heard of this before, but RNA polymerase, when it's making more copies of RNA, has a very high error rate. So a lot of mutations are naturally introduced into our RNA. And you might be concerned like, oh, that sounds really bad. But actually mutations are not always bad. Sometimes they're good. This is how our cells adapt. This is how we evolve. So not all mutations are bad. Just remember that. So when flu needs to make more of its own RNA to propagate itself, it uses our RNA polymerase. And so that means that just naturally occurring mutations are put into that RNA code. And this results in antigenic drift. So if you remember from the last video, is that when we look at the flu, we have the envelope with all the different spike proteins on it, which is composed of N and H proteins. And say we look at this H1N1 strain, and we looked specifically at the H1 spike protein. And if we zoomed in to this protein, we'd see its amino acid structure. It'd be a chain of amino acids. And we're just going to designate randomly that this little piece, that sequence of amino acids right here, is our antigen 1. So because when influenza makes more of itself or uses our host machinery to make more of itself, natural mutations are inserted into that RNA genome. So here's the first year. Over the course of the first year, those natural mutations, and I'm going to represent those by 2 X's here, a few mutations are put into that RNA genome of the flu. And if we remember back to Intro to Biology, we know that RNA codes for protein. And so this RNA genome codes for those H and N proteins that are on the surface. So if there are mutations, even little mutations in the RNA code, that means that there are going to be little mutations in our H and N proteins. So for example, I put little X's here at the H1 protein to indicate that it's slightly mutated. And so this does not mean that there's been a drastic change in the H protein, so we're still calling it the H1 type. So it's not reverted to or switched drastically to say H7 or H9. No, it's still H1N1, but we say that it's a slightly different strain of H1N1. And so if you remember that the Spanish flu, or the big epidemic of flu in 1918, was H1N1. And there was also a huge epidemic of H1N1 in 2009. Even though these are H1N1 overall class, these are not the exact same flu viruses. We call them different strains. And if you look at our PowerPoint, I've put a couple of designations of strains. So we'll usually call strains by where they were first isolated by the year. So for example, we might see California 2009 H1N1. There actually might even be more additions to specifically designate that strain. So that type, that strain, is very different from the 1918 flu, but it's similar enough that we still classify it as H1N1 in general. So again, getting back to this, we're here at year 2, so we have just a couple of mutations in the RNA code, and that led to a couple of mutations in the protein H1. And so remember, our one B cell is making our one type of antibody that was good for our original antigen. However, in year 2, our antigen has slightly changed so that that sequence of amino acids is slightly different. It's different enough that our original antibody, down here if you can see, our B cell 1 and our antibody 1 are no longer going to be able to recognize and bind or attach to this antigen. So now we need a whole new B cell 2, to make antibody 2 to recognize this new antigen. And over the years, this keeps happening. So year 3, we get a couple more mutations, and this leads to more mutations in the H1 protein. And that means that antibody 2 can no longer recognize this new antigen. And so we need B cell 3 to make antibody 3 to recognize this new site. So as you can see, there are just slight mutations every single year that makes it impossible for that old antibody to recognize this new antigen. And so this is why we have to make and administer and get new flu vaccines every single year. The WHO has a global campaign that starts every single year. I think it's, when do they start? I think it's in January or February, I could be wrong, but they start looking up, okay, based on our models, what are going to be the most prevalent and health concerning strains of the next year? So they predict, and then they make a whole new vaccine to the new strains, to the new antigens. They go through this very quick process, and I think it's a whole total of 9 months that they go through this process of selecting the strains, making the vaccine, and then getting it out to people. So really important. So 2 reasons why we need to get a flu vaccine every single year. 1, antigenic drift, these slight mutations that make the antibodies from last year no longer able to recognize the new antigens. And then 2, which we haven't talked about, is because for reasons that we don't really understand, we have a waning memory response to these antigens. So that means even if we had the same antigen, so antigen 1, antibody 1 memory response for some reason starts to wane after about a year or so. And we don't really understand why, for example.